So welcome back to the last recording for this chapter. Let's quickly talk about the concept check that I left you with at the end of the last one, and then we'll move on. Compare and contrast aerobic and anaerobic respiration. Include a brief description of the processes involved. Okay, so aerobic respiration involves glycolysis, the citric acid cycle, and oxidative phosphorylation. It results in the complete chemical disassembly of glucose so that you release the 6 carbon dioxides from glucose. It results in a total of 30 to 32 ATPs as a consequence of the whole pathway. But it requires 2 ATPs to get it started. In anaerobic respiration, if we're thinking in terms of glycolysis, and perhaps I ought to be a bit more specific about glycolysis, then we're talking about an investment of 2 ATPs, a net yield of 4 ATPs. We break down the glucose and get back 2 pyruvates. We get 2 NADHs, but we have a limitation in that we cannot run it continuously unless we combine it together with fermentation as an alternative way to oxidize the NADH back to NAD plus to allow glycolysis to continue to produce the small amount of ATP, the 2 ATPs that are produced as a product of glycolysis. If we're thinking about anaerobic respiration in terms of using alternate acceptors of the electrons from the electron transport chain, then the energy yield is going to be more or less the same, but the byproduct is not going to be carbon dioxide, it's not going to be water. We may produce something like H2S, hydrogen sulfide gas, otherwise known as rotten egg gas, swamp gas, or sewer gas, or some other chemical compound as a byproduct of accepting the electrons. But that, again, allows for the buildup of a hydrogen ion gradient that can drive the synthesis of ATP. So those are the sorts of things you ought to be thinking about in terms of this particular concept check question. Anyway, let's get on with the last section, thinking about how versatile cell respiration is, its flexibility, and then how it's regulated. So one of the things that we always need to remember is that while we may write these overall equation for cell respiration, focusing on glucose as their primary fuel, and that is in fact the case for cells, we should not lose sight of the fact that any type of food molecule, whether it be amino acids, proteins, or fatty acids from fats, lipids, they can also be used as a source of energy. And in fact, if we think in terms of the gram for gram energy content, and I think I've given you these probably given you these numbers before, but it never hurts to remind you. If we think in terms of carbohydrates, 1 gram of carbohydrate is equivalent to 4 kilocalories. 1 gram of protein is also equivalent to 4 kilocalories. 1 gram of fat is equivalent to 8 kilocalories. Some people say 8 to 9. My feeling is that the vast majority of people would say 8. So somewhere here, we must also recognize why fat can be so energy dense. There must be something about the way fat is processed in cell respiration that allows us to account for that energy density. Excuse me. So this diagram shows the way in which the different components of food are processed. So in the case of carbohydrates, any sugar that can be converted to either glucose or fructose can be processed directly through cell respiration. So if we're talking about glucose or fructose, they can enter directly into cell respiration via glycolysis. If, however, we have sugars other than glucose or fructose, so long as we can convert them into one of the two, we can process them into glycolysis in some fashion and get the energy yield out of them. So carbohydrates are relatively easy to process. In the case of proteins, once we break them down to their constituent amino acids, the next step is to get rid of the amine group. Because the amines, it turns out, are toxic to some of the enzymes in cell respiration. So it's really important to get rid of those amine groups, and those are excreted as uric acid and urea in our urine. They're filtered out by the kidney and by the liver, or in the liver, the amine groups are removed and then passed on into the bloodstream and filtered out as urine, into the urine as uric acid or urea, and excreted from the body that way. So if you're eating a lot of protein, you have to be very careful because your kidneys require water to function. And if you're eating a lot of protein, you're asking your kidneys to do a lot of work. So it's not that protein is necessarily bad for you in larger amounts. It can actually help you lose weight so long as you don't go overboard. You keep things in moderation. Remember, all things in moderation. But if you eat protein, you have to make sure that you drink enough water to keep your kidneys active. If your urine is constantly a straw color or an orangey color and it smells kind of funky, it simply means you're eating a lot of protein but not drinking enough water to keep your kidneys functioning normally, and you are risking kidney damage. So it's important to drink water. In the case of fats, the glycerol can be converted to G3P. Let's not write that in because I can see that I've got that already there. So we'll take that. Oh, I just erased everything, didn't I? No, I can put this in. Yeah, so that's from amine groups, and this is glucose or fructose is what we want. So the glycerol and G3P, they're very closely related. Glyceraldehyde is actually glycerol with a phosphate group attached to it instead of a hydroxyl group. That's the difference if you're curious. Fatty acids are broken down in the liver by something called beta oxidation. Excuse me. And that produces acetyl-CoA groups. So an average fat or fatty acid that can yield up to 8 to 10 acetyl-CoAs, and remember glucose only yields 2 acetyl-CoAs. So immediately you start to see why fats are so energy dense, because they make acetyl-CoA, or they yield acetyl-CoA, which enters the citric acid cycle, which is where you get the vast majority of the NADHs and the FADH2 being produced. Remember, only a little bit of that comes from pyruvate oxidation and from glycolysis. The vast majority of the NADH and the FADH2 comes from the citric acid cycle. And so the acetyl-CoA feeds into the citric acid cycle. And so by producing a lot of acetyl-CoA from fatty acids, you feed the citric acid cycle, which increases the amount of NADH, which increases the size of the hydrogen ion gradient, which increases the amount of ATP you can make. The fats are very energy dense, which is the reason why fats represent a lot more calories and also represent the most efficient way for an animal to store excess calories, because they represent a lot more calories when processed because of this production of acetyl-CoA. Now, ironically, not only can we process all sorts of food molecules through cell respiration, the cell respiration pathway can also serve as a source of raw materials for making the different types of molecules. So it turns out that really what cell respiration is doing is running on a very fine edge, depending on the cell's energy budget versus its carbon budget. If it needs ATP, if it needs energy, then the focus is on making ATP. However, if it's able to make enough energy to keep itself running, but it needs organic molecules in the form of amino acids or fatty acids or sugars, then you can take some of the molecules out of cell respiration that would normally be the byproduct of proteins, fats, and sugars fueling cell respiration, and literally run those reactions in reverse, with the exception of glycolysis and to some degree beta oxidation. There are alternative pathways that allow you to do those, run the chemistry in reverse, not the chemical reactions, but the chemistry, and be able to make these raw materials. So if you want to make amino acids, you pull some of the intermediates out of the citric acid cycle. So let me put this in here. So an old name for the citric acid cycle is the Krebs cycle, named after Dr. Hans Krebs, the English biochemist who led the group that figured this out. He got a Nobel Prize for that, by the way. No surprises there. But the modern name is citric acid cycle, but an older name is the Krebs cycle. So you can pull some of the intermediate molecules in the Krebs cycle out and use those to make amino acids, because the amino acids can in turn, with the removal of the amine group, be shuttled into the citric acid cycle as those intermediates. If you want to make fats, well, you take the acetyl-CoAs, link them together to make the hydrocarbon chains, add the carboxyl group to the end to make the fatty acids, take some G3P. Remember, G3P can be derived from glycerol. So you can reverse that, make the glycerol and make your fatty acids. And so if you take in excess calories in the form of protein or sugars, guess where those go? Guess where they wind up? You guessed it, fats, because we can turn these excess products out of the citric acid cycle, out of what is referred to as gluconeogenesis. Which is the pathway that allows us to make glucose from pyruvic acid. We can take G3P out of there and use it to make glycerol and store fat. So if we've got excess calories, that's why they're stored as fat, and this is how we can make them as fat. Of course, if we need sugars, pyruvic acid or pyruvate can be converted to G3P, which in turn can be turned into glucose as necessary and stored as glycogen if we need to. Because if we deplete our glycogen stores, we need to make glycogen. If we don't have enough carbohydrate to make glycogen, we can cycle it out of other things. This is the reason why, by the way, the triple threes exist. in health sciences. Remember, 3 minutes without oxygen, 3 days without water, 3 months approximately without food, because your body can self-digest its muscles, its fat content, and use those to make the sugars necessary to drive cell respiration, to keep the brain and the heart functioning, the primary organs functioning. And it will literally eat itself in order to keep the primary organs functioning. And it's very, very good at doing that, and becomes extremely calorie efficient at doing that. That's the reason why starving yourself usually does not lead to dramatic weight loss after a few weeks, because your body simply becomes very much a calorie miser in the way it processes energy. But it can use itself as a source of energy, and that's why when you see pictures of people who have been calorie depleted, so think in terms of the pictures of concentration camp survivors or people who have eating disorders. The reason why they look like they are skin and bones is because essentially that's what they are. Their bodies have digested all the fat, digested all the muscle. And by the way, it will favor muscle first before it favors the fat. The fat it views as its long-term energy source, so it will preferentially break down muscle before it breaks down fat. That's the reason why, if you are dieting and cutting down on the number of calories that you take in, that you make sure that you provide a constant source of amino acids and to keep the muscles built up, and that you constantly exercise your muscles, do strength exercises to force your body to maintain the muscle. Because if you are reducing the amount of calories that it gets in, or playing with the combination of foods, your body will redirect those resources, and it will start to break down muscle in order to repair muscle or to build up glycogen if you have reduced the amount of carbohydrates that you're taking in that could be readily used to make glycogen reserves. The body will outthink you and outsmart you and out-chemistry you every time, because it's that way. That's how it functions. And unless you really understand how all of this works and how the whole system interplays with each other, you can dig yourself into a deep hole. You can get into a whole lot of trouble very easily by playing with the foods that you provide your body with. The end point, or the end result of all of this, is a simple recognition. At the heart of everything that a cell does is the cell respiration pathway. It's a source of energy, it's a source of raw materials. And so as a consequence, it needs to be tightly regulated. And you may recall in the first animation for this, or the first recording for this chapter, I pointed out that in glycolysis, the first irreversible step is the utilization of that second ATP, where you add the second phosphate group to a fructose to get fructose 1,6-bisphosphate. That chemical reaction, catalyzed by an enzyme called phosphofructokinase, is irreversible. You cannot go backwards from there. And so if you want to slow down or stop or speed up cell respiration, that's the step to focus on. Because you can either prevent cell respiration or slow it down by downregulating the enzyme at that step, or you can speed things up by upregulating that enzyme, because that's the key step in cell respiration. So not surprisingly, the inhibitors of that enzyme turn out to be products associated with cell respiration. So surprise, surprise, ATP itself turns out to be a negative inhibitor, a negative regulator of phosphofructokinase. It binds to phosphofructokinase in such a way through an allosteric site that the enzyme changes shape and no longer functions. It's an allosteric inhibitor, non-competitive inhibitor of phosphofructokinase. So when the cell has enough ATP, the ATP feeds back on phosphofructokinase and downregulates it, so that It's not excessively making ATP. Now, I know that seems weird, excessively making ATP, but remember, all the cell is interested in is having enough ATP to maintain its activities. Remember also that cell respiration, or the chemicals of cell respiration, can be used for other purposes. So if you've got enough ATP, then you can start utilizing those products for other purposes. The citric acid or citrate intermediate in the citric acid cycle also serves as a down regulator of phosphofructokinase. And the purpose for that is to keep the glycolysis pathway and the aerobic portion of cell respiration in the mitochondria in sync. If there is too much citrate, meaning there is too much pyruvate being produced in glycolysis for the mitochondria to handle, there is no point in feeding more into it. So you want to downregulate phosphofructokinase to downregulate glycolysis so you don't have as much pyruvate feeding into the mitochondria. Keeps everything in parallel and in sync. Now, we also know that we want to upregulate cell respiration. So the classic way of doing where that's required is in the fight or flight response to adrenaline. And adrenaline, when it binds to the receptors on muscle cells and other cells that, like the liver cells, that contain a lot of glycogen and can be processed to produce a lot of energy, trigger the production of a molecule called AMP, which is the third member of the ATP, ADP, AMP family. And AMP, it turns out, is an enzyme stimulator or upregulator. It's a control molecule, and it controls other proteins. And in the case of phosphofructokinase, it's an activator. It upregulates phosphofructokinase. It upregulates cell respiration. And so when you get the adrenaline response, all of that energy that's being produced to allow you to do the fight or flight response, that's a product of AMP upregulating phosphofructokinase and turning up the thermostat, so to speak, on cell respiration. So you produce that huge amount of ATP necessary for the fight or flight response. That is perhaps the key indicator as to why cell respiration is so important. And I used to have a chart that was provided by a chemical supply company that showed all the known biochemical pathways in all different cell types: plant cells, animal cells, fungal cells, bacterial cells, you name it. If it was a chemical pathway that was known, it was represented in that diagram. And at the heart of that diagram, and it was a 6x4 poster sheet, was the cell respiration pathway running down the center of it. And essentially every biochemical pathway on that 6x4 sheet, all cellular chemical pathways, tied to cell respiration in some fashion, feeding into it or feeding out of it in some fashion. That's how important cell respiration is. And that's the reason why it needs to be regulated as much as it is. And it is very, very tightly regulated, because not only is it a source of ATP for cellular work, it can also serve as a raw material for the organic molecules that cells require, the amino acids, the sugars, the fatty acids, and so forth, that become the building blocks of cell. So a final concept check, because that's the last slide of the presentation. Why do fats represent a better source of energy than carbohydrates? Under what circumstances would your body make fats? Well, fats represent a better source of energy because they produce a lot of the acetyl-CoAs, more acetyl-CoAs per fatty acid than per glucose molecule. And because it's the processing of acetyl-CoA molecules in the citric acid cycle that yields the greatest number of electrons in the form of the NADHs and the FADH2s, that then yield the greatest source of ATP as a product of cell respiration. And that means that fat can make a greater contribution to the overall ATP content of a cell than a carbohydrate. It's not a direct correlation to the number of ATPs, because remember, fats are only twice as energy dense as carbohydrates. So it's not a direct correlation to the increase in the number of CoAs, of acetyl-CoAs. But there is nonetheless a correlation to the increased number of acetyl-CoAs from an average fatty acid versus the number of acetyl-CoAs per glucose or per carbohydrate. Under what circumstances would your body make fats? Whenever you have an excess of calories that cannot be consumed by the body, then those will be redirected to the formation of fats. And we saw how that is a product of cell respiration. The fact that excess organic molecules from amino acids, excess organic molecules from carbohydrates can both be converted into sources of the building blocks for fats. And so any excess calories that come in the form of those molecules can be in turn directed into the production of fats for storage against the needs in the future. So that's cell respiration. Hopefully there's been a sufficient explanation here, and I know I've gone into a lot of details in some circumstances, but I want to make sure that I try and answer as many of your questions as possible, or potential questions. Again, if there are any comments, questions, or concerns about anything that I've covered in any of these presentations about cell respiration, by all means, feel free to come and see me during office hours, and I'll be more than happy to try and clarify these for you. So until next time, when we cover photosynthesis, let me know how you feel about this new approach to the recordings, if having it broken up into multiple smaller recordings is a better way for you to approach the material. I'll be interested to find out. Otherwise, I'll see you back here again when we cover photosynthesis. Bye for now.